organizing this conference and inviting me to speak. I will first share a couple of slides that irreplaceable Judy Campisi provided to me many years ago, over 10 years ago, and which remains still relevant. So the first slide demonstrates or illustrates that with age, at a certain point, there are many diseases that rise exponentially. So therefore, there must be basic processes that drive aging, and most of, or perhaps even all, age-related diseases. Therefore, the conclusion from that is logically that treating basic aging processes can prevent or attenuate or possibly even reverse such horrible age-related diseases as neurodegeneration and memory loss, degeneration of other tissues such as osteoporosis, sarcopenia, and frailty, vascular dysregulation, metabolic dysregulation, and even cancer. So Judy pioneered these thoughts quite a while ago. In our collaborative work with Professor Judy Campisi and her former postdoctoral fellow Oki Jeon, who now is professor in South Korea, we kind of explored this idea or possibility that aging is not a decline of proteins, vitamins, and so forth. Aging is, in contrast, a molecular excess. And therefore, senescence that used to be thought to be intrinsic cell autonomous process where senescence or damage to cells accumulates over time can be rapidly induced in completely young animals quickly in a couple of weeks when they're still young by heterochronic blood transfer or by transfer of old blood, 50% of old blood into young mice. And this work was published a couple of years ago, and it shows that this heterochronic transfer of blood rapidly induces senescence of multiple organ systems such as liver, kidney, and muscle, and ages and senesce the entire animal. Furthermore, this can be also observed in vitro when young cultured cells are exposed to young versus old human plasma. There is induction of SA beta-gal, and looking in vivo in animal muscle, one can clearly see rapidly induced SA beta-gal positive cells in a satellite or stem cell position. More importantly, exploring this paradigm in more detail, we found that old serum dominates over young when they are mixed. And I put exclamation point here because, again, it then poses this very interesting question. If old sera is dominant, old circulatory milieu is dominant, how can one rejuvenate an old animal or old person by adding a little bit of young blood or fractions or factors? So what you can see here are the cells that are cultured with young serum, old serum, or a mixture 50-50 of young plus old. And for P21, old serum dominates over young. For P16 is the same outcome. For IL-6, another SASP component, the same outcome, old serum dominates. For MMP3, it dominates. For lamin B, one, old serum dominates, there is decline expected of senescent tissue, and cells do not proliferate well. Once again, old serum dominates over young. So that introduces an interesting point that in heterochronic parabiosis, where old mice were rejuvenated by suturing to young animal, there could have been many factors, including organ sharing and environmental sharing, but one of the factors was clearly removal of age-elevated systemic proteins by the young mouse. And that was the most likely reason or effect on rejuvenation. Then parabiosis, of course, is not very elegant or well-controlled experiment because animals are sutured. And some time ago, we developed small animal blood exchange where mice are not connected to each other. And that experiment again showed that old serum dominates over young, for example, in reducing the number of neural progenitor cells that are located in the hippocampus and then to gyrus here. From this level in young animals, and these animals were also injured in the skeletal muscle to that level. So old serum really has deleterious effect on formation of new neurons in the area of the brain that is responsible for memory and learning. On contrast, young serum does not seem to be rejuvenative in this clean experimental setup. So to our surprise, people still interpreted even this paper as something where old person needs to sneak into the blood donation clinic and steal the young blood, even though our result was quite reciprocal, right? That one would need to attenuate or remove age-elevated proteins from circulation. So then the next setup was even better developed. A better developed setup was then introduced next. And here, one does not need to exchange blood between two mice, but can simply dilute old plasma by using saline albumin. And you see the setup here on the left. So this is plasma dilution setup. Once again, all of these are much, much better than heterochronic parabiosis, and they allow to do much more. And so plasma dilution turned out to recapitulate every single positive effect of heterochronic parabiosis, but stronger, such as that now old animals that underwent this procedure, which we called all neutral blood exchange, since there is no age to the blood, which simply dilute plasma, now they are cognitively are the same as young animals, and this is in test of novel object recognition, and novel texture recognition. And they are statistically the same, although there is still trend to be lower in the hippocampal neurogenesis in the subgranular zone of dentogyrus. This is different from heterochronic parabiosis. By the way, this data is back from 2004. It was attempted by us to be published together with muscle and liver, but the neuroscience reviewers gave us hard time, so we shared these unpublished results, which were reviewed in Nature twice with Tony Weiss-Corey, and fortunately they took it forward and got it published also in Nature in 2011. And so what you can see here is that there is a decline in young animal that is sutured in parabiosis with the old. But if you look at the scale, the decline is quite small. And there is also improvement in the old mouse, but if you look at the scale, the improvement is much smaller. And old mouse after parabiosis is still much, much worse than young mouse, but not after neutral blood exchange. After neutral blood exchange, old mouse is statistically the same as young mouse. So once again, the conclusion here would be that aging is excess of systemic proteins, and we can reverse age-related diseases by their dilution or clever attenuation. So then looking at systemic proteins in more detail, first, if you focus on the signal transduction networks, there are many proteins that become upregulated with age in old people. And after rounds of therapeutic plasmapheresis, which is kind of the same as neutral blood exchange, so old plasma is being gradually diluted, that age elevation of systemic proteome becomes diminished and made similar to that proteome of young people. So you see kind of a dilution of aging, looking at the antibody-array proteomics. And another interesting observation here on the right is that if you look sometime later, after you drop down the levels of age-elevated proteins, you see that all of the youthful proteins are now came back. So they are increased in mice and they are increased in people. So it is quite possible that once age-elevated systemic inhibitors of canonical signaling pathways are diminished and calibrated to the youthful levels, we do not need to add young factors because they naturally come back. They come back from derepressed gene expression. And in people who underwent rounds of therapeutic plasmapheresis, as shown here in the schematic, that is accompanied by rejuvenating circulating blood subsets, which after each round have less damaged DNA and are less senescent. Each dot here is a round and each line is a person. And moreover, the ratios of lymphoid to myeloid cell is now rescued. And this is super important because with aging, myeloid or inflammatory cells prevail, and lymphoid cells, which are important for combating viruses, for example, and cancers and other infections, become diminished. So this ratio is restored. So not only systemic proteome is recalibrated by transient and quick plasma dilution, but also that is accompanied by functional responses in blood cells. And in animals also by improved regeneration and function of multiple organs. So this system can be also used to measure biological age. What is shown here in the cartoon, that we can predict many things using population statistics. For example, we can predict people's weight using population statistics of what they wear or what they eat. But we designed the method to actually precisely measure biological age through proteome noise or standard deviation, and uncovered certain noise detectors, shown here on the right, that standard deviation in this is low in young and become increased in old, and is reduced significantly by the rejuvenating procedure of therapeutic plasmapheresis. And so biological age could be measured. And biological age based on this measurement of protein noise is reduced rapidly after procedure of therapeutic plasmapheresis and is increased by diseases such as mild cognitive impairment. Moving on, we became interested in understanding what are those clocks. You probably have all heard about DNA methylation clocks and how they are different from conventional clocks. And so they are different because conventional clocks are real and they operate in our real-time space continuum, but biological clocks are a mathematical simulation, which is illustrated here in this cartoon on top part. So furthermore, when one looks at the DNA methylation array, there is no linear progression of aging, and people do not neatly cluster into young and then eventually old. So this is the raw data or primary data analyzed by T-SNP. So how do we go from this clustering to this nice straight line, which you probably have seen in DNA methylation clocks? That happens because we modify values by our biologically arbitrary weights that are mathematically very important, that they allow us to simulate this straight line progression. So there are many positive things about biological age clocks that are based on this mathematical simulation by biologically arbitrary weighting the data. But the negative thing is that there might be various progression curves of how we go from being young to becoming old. For example, increase in aging, then healthy plateau, and then additional increase. And they are all erased in favor of the mandatory mathematically simulated straight line. So the first demerit is we don't know how aging progresses really. But once we have established this linear progression, we can predict things very accurately, and the things that we are predicting, they don't have to be connected or causal. They don't even have to be physically linked to what data we are using. For example, we showed that one can predict how many people live in the United States over years from the DNA methylation array of few participants. And because people who live in the United States basically is the reflection of progression of time, then using exactly the same clock, one can predict ages of people who are living in the United States today or living anywhere in the world. And the same principle is true about first generation clocks or next generation clocks, which are listed here, including pheno-age, grim-age, mammalian cause-age, etc. Their main training variable remains progression of time, regardless of whether it is called health score or ages to death and so forth. And because of this, these mathematically simulated clocks, which feed the data mathematically and against biological knowledge of why do we feed data like that. They work very well as predictors, but the biological relevance remains less known. And once tested on several different experiments or data sets, it turned out that they have errors of prediction. For example, this thin line is perfect prediction, and they have errors of prediction throughout the whole chronology. And furthermore, they do not distinguish healthy people from patients with various diseases in their predictions. And the errors in age determination for the same person could be as large as 10 years or more. Additionally, the features that clocks are using do not seem to be biomarkers. So this picture with dots, each dot is individual beta value or feature that machine learning models have selected to predict our age. And you see that for many of them, there is no correlation and DNA methylation change with age. They're all here at the bottom. And once all of these predictors are thrown out and machine learning is repeated, the next predictor is built and it is working very well, but a little bit worse than the first predictor that was the best one. But now you see that there are almost no dots at the bottom, so each feature has a little bit better correlation and DNA methylation with age. So interestingly, even though DNA methylation assay has been used for training the predictor, it doesn't seem that the predictor is working on analyzing changes, epigenetic changes with age. It is doing something else, which is very interesting what it is. And then to resolve this problem, we shifted from the levels to the noise. In our hypothesis, noise is the biomarker of aging and disease, or standard deviation, or absolute distance from mean. And this is represented by this one cytosine, one area of the genome, where each dot is a person, and starting from young adulthood, like 25 years old, the levels of this particular DNA methylation of this area of the genome are on average the same, but the noise in people becomes larger with age. And so that might be the answer as to why biomarkers of aging were elusive, because the levels are masked by the individual variation in genetic environment and also become very variable as we grow older. So instead, if one uses standard deviation as the metric of aging or noise or biological entropy, then we can see what the linear progression, what the non-linear progression of aging really looks like. How do we age? And we age by increasing noise, then we have plateau, additional increase, and eventually exponential increase in biological noise or entropy. The difference between health and disease becomes visible. We can identify that as much as 1/4 of all tested cytosines in our genome, out of 5,143,000 cytosines, become noisier with age and go to pretty much similar progression. This is how we grow old. This is young, and then we grow old. And then we can see difference between health and disease, but looking at the entire data set, there is no rhyme or reason, so one needs to identify noise detectors. These are the optimal subset of 460 cytosines, which allow us to see progression of aging through biological noise, and it is visible even at the single cytosine level on individual cytosines in which we also have identified. So overall conclusion is that as with any measurement, quantification instead of simulation or prediction of biological age opens many possibilities for testing rejuvenative strategies, medicine, and so forth. Metaphorically speaking, if there were no scales, people could have delusions whether they lost weight or not, but with the scales, they can develop a better plan and lose weight more consistently. So scales work on the concept of gravity, and noise barometer works on the concept of biological entropy. I think that I will stop here and just, there are many thanks to people who worked in the laboratory, particularly Serafino May, Connor Llewellyn, and Joshua Blanchard, who worked on the development of noise barometer and the tests of conventional simulation models, and everybody in the lab, Dobry Kiprov, Joel Kramer, our notable collaborators, and many thanks to our funding sources. Thank you very much.